I'm very pleased that we have this opportunity to invite Professor Christina SMOLKE from Stanford University. So before I give the stage to Christina, I want to mention a few things about her. So first of all, actually, this is not her first time visiting HKUST, because around 11 years ago, actually, this campus, we hosted a very large synthetic biology conference, SynBio 4.0, together with many distinguished scientists, Chris Voigt, Drew Wendy, Christina, and many others. So at that time, it was the biggest conference ever hosted at HKUST. So 10 plus years ago, we have this vision that SynBio is going to shake up the economy. So we are very happy that very recently the university is able to receive a donation from the Recoding Foundation. So the university is going to set up this Recoding Institute of Synthetic Biology. And although we have been kind of delayed for 10 years, I think there's still a lot of opportunities. And so I'm very pleased that we very much have the kind of the leading scientist, also entrepreneur to this campus. So Christina got her undergraduate around the same time I got my PhD from MIT. I think she got her undergraduate from USC around 1997. Then she moved on to UC Berkeley with Jay Kissling, and then chemical engineering for her PhD and period postdoc. And she got recruited to Caltech, and she did extremely well in Caltech. And then she decided to move on to Stanford. So she became a full professor several years ago. And then because she's very successful academically, and she also is very passionate about translating SynBio to making real industry impact. So recently she founded this company called Enzyme, right? Anyway, she will tell you the right pronunciation later. And this is a very exciting company. I'm not going to spend time to talk about it. And we'll let Christina to share with us about her research, her patient, and her company. And please join me to welcome Professor Christina SMOLKE.
So it's a pleasure to be here today and to be able to share some of our work with you. And I'm going to focus this talk on the work that we do in the plant natural product space. As was mentioned, a lot of the work that I'll share with you today has now been translated into a company, Enzyme, where we're working to commercialize the applications of this technology. But what I'll share with you today is the earlier foundational work that was done for the development of it in my academic laboratories at Caltech and then Stanford. Later on, actually, I have a second talk that I'm giving today, which will focus on a different side of the work that we do in the lab, which is our functional RNA engineering work. So this will strictly focus on the natural product work. And also because this is more of an introductory lecture, I wanted to start off and just give a little bit of context for how we think about synthetic biology and what it enables, and then how that feeds into some of the work that I'll share with you today. Okay, so in thinking about synthetic biology, I think we need to recognize, right, that it builds upon, I would say, the foundation of genetic engineering and recombinant DNA technology, which has been around for decades, right? And we can start there and maybe think about, just with sort of the basic technologies that came out through recombinant DNA technology and genetic engineering, what type of impact that has had. And so I want to just briefly introduce the impact that a single protein can have, right? And so this is a protein, cetylysin, which is actually used in laundry detergent to clean our clothes, right? And so previously, the original version of this protein would only function in high temperatures, which is why we oftentimes washed our clothes at high temperatures if we wanted to get them very clean. So researchers basically spent a lot of time using recombinant DNA technology and protein engineering techniques to engineer a version of this protein that would exhibit the same activity at lower temperatures, right? So what that then meant was that people all across the world could now wash their clothes in cold water, right? Or at least water that did not have to be heated up. And that then led to a very substantial decrease in heating, the heating needs, right, for heating up hot water. And you can see the translation of what that would be in terms of barrels of oil per day, which is quite substantial. So this is really just to highlight, again, the impact that engineering a single protein can have in the space of bioenergy, right? And you can contrast this savings to a few different things, right? So for example, if you just look at this savings, this is about 20x the savings or the amount of oil that was spilled per day in the BP oil spill that happened in the Gulf of Mexico. In addition, if you think about the impact that, say, biofuels was going to have, right, when people were trying to engineer microorganisms to make biofuels and feed them different feedstocks, the impact of this single protein, a much simpler system, was about 200,000 times that, right? So we want to think very carefully, again, about the types of systems that we build, what type of impact they're going to have. Sometimes simple it can be quite effective, right? And sometimes having more complex systems are also going to be quite effective. But this will segue then into just sort of laying out the foundation or the background for the tools that drove genetic engineering, right? And this, again, is sort of the first generation of biotech that came out several decades ago, when you had a set of core basic tools that was fueling applications in this space. And these were things like recombinant DNA technology, which allowed us to cut and paste DNA. These were things like polymerase chain reaction, which allowed us to amplify and make simple changes to DNA. And it was also things like DNA sequencing, which allowed us for the first time to be able to read out the genetic code. So these were all very basic capacities, but together they allowed for some very impactful applications. And in particular, I'm showing you two of some of the more notable ones, recombinant insulin production and recombinant erythropoietin production. Both of these applications basically took an organism. In the case of insulin, it was a bacteria, E. coli. In the case of erythropoietin, it was a mammalian cell culture, right? But each one of those cell cultures were basically engineered with recombinant DNA to express a single recombinant protein, okay? And that recombinant protein in either insulin or erythropoietin was the final product. So they used the cell as a manufacturing platform. They could then make that single protein product more efficiently, purify it, and then use it as a therapeutic. These led to the development of what we now know as very large pharma and biotech companies, Amgen and Genentech, right? Started with these sort of core examples. And one thing to note here is that both of these examples are fairly simple, right? A single gene into an organism, and that protein product is the final product that's going to be applied. So then the question, I think, that the field began to look at was, okay, these are sort of applications that we have when we're talking about maybe a system with a single protein as the product and a single gene that we're inserting into a cell. What can we do and how can we advance the space if we can expand the number of genes that we can put into a cell, right? So instead of one, we go to 10, we go to several tens, we go to hundreds, we go to thousands and entire genomes. What is that going to open up and allow? If instead of just making a single protein product, what happens when we can begin to make complex chemicals and materials? And then finally, can we move into a scenario where the organism itself is the product, right? And all of these things are things that are being realized today. And so those sets of questions basically move us into the realm of engineering complex biological systems, where you have many interacting parts and many interacting pieces that have to come together to give you an overall functioning system towards a particular objective. And, at least the field of synthetic biology, and this field of synthetic biology, that's sort of how it's framed oftentimes, right? And if we think about this, then, you could think a lot of times there are analogies drawn to other engineering disciplines where the frameworks for engineering systems was more well laid out, right? So we can take the example, in this case of the Mars rover, where you're engineering a system that is going to go into a complex environment You want this system to be able to receive signals from its environment, to be able to process that information, and then ultimately be able to make decisions about what actions it wants to take, right? And you can develop a well set of kind of standard parts and pieces that will come together and fit together to give you this functioning system. And you can ultimately break these down into different functions, such as your sensors, which are going to receive the inputs, your actuators, which are going to be the action that the system takes, and then ultimately the circuitry, which connects the two, right, and makes decisions. And so synthetic biology took this as sort of the inspiration to ask, you know, if we want to develop biological systems that can do the same thing, that can basically receive information from the environment, process it, and then make decisions about what actions it could take, we have to develop similar frameworks and components that will allow us to program similarly, but now within a biological system, right? But very similar in terms of, at least at a high level, what types of functions we wanted to be able to build. And so really then, that is how I view synthetic biology, right, and its contribution to the space, is that largely it is a field that is focused on tool generation to support the engineering of complex biological systems. And so you had a second generation of biotech tools that were built upon the first generation that advanced these capacities, right? These are some of the early ones that have come out over the past decade, right? You had synthesis, so the advances in DNA synthesis, which now allow us to be able to synthesize entire genomes from scratch and assemble them in organisms. That's important because it allows us to basically decouple design and fabrication, right? Whereas before, we were very limited in sort of the de novo synthesis or from scratch synthesis of DNA that we could design. Abstraction, right, is sort of a framework that allows us to take a complex system and abstract away complexity so that we can focus on the layers that we want to engineer. And there have been many kind of frameworks for proposing how to think about abstraction in biological systems that have been useful in this context. And then standardization, right? The idea of really thinking about, as we define and develop these biological parts that we're going to use in particular cells, how do we begin to standardize, right, their definition, their composition, so that we can move them across different systems and still have expected behavior? And how should we think about standardizing characterization and reporting so that that type of information can be well understood across laboratories? All of these are really meant to be able to scale, right, how we engineer systems and to be able to coordinate across multiple researchers, laboratories, institutions, et cetera. And so now the idea is that as these tools, and what the field you've seen sort of been able to begin to ask, is that as these tools come online, how does that advance our ability to engineer biological systems, and where does this take us? And what you'll oftentimes see within the space of synthetic biology is what they call this engineering design cycle, which essentially is this cycle that goes between design, build, and test, right? You design a system, you then test it, you then build it, and then you test it, you characterize it, gather the data, and in almost every case, your first design is not going to work the way that you want it to, and so you're going to iterate within this loop, right? You take the information that came out of your first design, and then you make a next generation design. And you can see then how the tools of synthetic biology basically fuel the different parts of this, right? In terms of abstraction, synthesis, and standards. And ultimately, within the field of synthetic biology, you see different strategies about how to advance this cycle. So there are strategies that allow us to go around this cycle, this circle faster, right? So just complete cycles faster each time. There's also strategies that allow us to go around more times in parallel, right? So the high throughput strategies. So instead of doing one design and then a second design, you do 100,000 different designs, characterize them, and then use that information to fuel the next cycle. There's some strategies ultimately that try to get us to go around the cycle smarter, right? So as we do this again and again, how can we take the data and basically use different computational strategies to be able to learn how we should improve our designs, right? So that we have a better starting point as we go forward with different applications. And you see all of that coming out in the space. Okay, so with that is sort of the introduction to synthetic biology, right? And the sort of growing tool base that is fueling the field of biotechnology. I think the next question is, as we have this increased capacity to engineer biology, what problems are we going to choose to work on, right? Where are we going to gain the most value from this technology? And that's going to then lead us into the next part of the talk, which is going to focus more on the research that we've been doing in our laboratory. And so I want to start off by getting us to think a bit about what biology does and what biology is good at. As someone with a background in chemical engineering, I really think about biology as a system that makes molecules, right? That's essentially what biology has evolved to do. It's basically making molecules. Those molecules interact in different ways, and those interactions basically give us different behaviors, right? And the molecules that biology makes is being crafted by evolution, right, to fundamentally have properties that allow those molecules to interact with other biological molecules, right? That's essentially how this works. And we in particular are very interested and continue to be interested in the types of molecules that plants make, right? And if you think about plants as chemists, what we know is that they really make an incredible diversity of very interesting, unique, and high-value compounds, right? And from what I mean by interesting is that they have very unique chemical structures and very unique properties that have oftentimes been very difficult for us to be able to replicate and reproduce via synthetic chemistry methods, right? And you can imagine that there's an evolutionary pressure on plants to be able to do this because they are stationary objects, right? And so they basically can't run away or move themselves, right, to different locations. They're sort of set in one place. And so the way that they interact with their environment is by making molecules, right, that will either act as defense, protection against different threats, that will recruit different organisms and microorganisms to them, right? But that's essentially their way of interacting with the environment. And so again, they've been under evolutionary pressure to do this type of biosynthesis. And so it's not surprising then that we have, over the centuries, we have basically looked to plants as chemists for providing useful molecules. And this is certainly true in the space of medicines and therapeutics, right? Starting with traditional medicines, right, where it was recognized very early on that plants have healing properties, right? And then in many cases, as we sort of transition to modern medicine practices, being able to identify the active ingredients in plants and ultimately isolate them and use them in formulated products. Now, you can imagine then that when we rely strictly on nature to make molecules, especially in the context of therapeutics, and to discover new medicines, there are a number of challenges that come up, right? The first is that you have limited scaling capacity. Plants usually make very, very small quantities of these compounds relative to their overall biomass, right? And so if you're using the plant to sort of recover and isolate the compound and then using it to sort of scale production for drug development, that can become very limiting. Also with the slow growth cycle of plants, right, it can also become very limiting. The other thing is that in some cases, this has led to over-harvesting of rare medicinal plants, right, which ultimately can lead to loss of biodiversity, right? And that's, again, an issue in the broader context of how we think about our resources and how we leverage them. The next challenge is that in many cases, although these compounds can be very useful and interesting lead compounds as a therapeutic, we oftentimes want to further modify them to improve their properties, either increase their efficacies, their bioavailabilities, or decrease side effects, right? And in practice, that can sometimes be very challenging because of the complex nature of these structures to be able to sort of fully access different modifications on top of that complex structure through synthetic chemistry means. And then finally, if we think about the entire potential chemical space that exists, right, and all of the molecules that we would like to be able to develop through these types of strategies, it's not going to scale, and it's going to be impractical, right, to really fuel an advanced drug discovery this way. Now, even with those challenges, right, it is the case today that plant natural products play a very central role in global health. If you look at therapeutic space, right, and small molecule therapeutics, which even today comprise 75% of the market, the vast majority of small molecule therapeutics are natural products, right, or derived from compounds that are natural products, and at least half of them come from plants. And so they play a very essential role. Furthermore, if you just look at the overall global use of plants in healthcare, 2/3 of the world uses plants as their primary source of healthcare. So this really plays a very critical role. Now, the World Health Organization has a list of essential medicines, which basically means they identify these as being essential for kind of basic human health, right, and quality of life. And 20% of the compounds on that list are basically plant-derived compounds. And so with many of these essential medicines, they actually track access, right? And the medicines that are used in palliative care is an example of this. And in many cases, access is a big issue, right? And so with these classes of medicines, most of the world's population, over 5 billion people even today, have insufficient access to these medicines. And this is for a number of reasons, but within the top sort of 4 or 5 is certainly cost and just hurdles and barriers with being able to access the raw plant material to produce these compounds. And so taking this as sort of a segue, if we think about what a supply chain that is based upon growing plants looks like, this might then key into what some of the issues are, right? So this is what a supply chain based upon plant production looks like. This is a picture of an opium poppy field in Tasmania, which supplies about half of the world's legal material from poppy to make a variety of different medicines. And so you can imagine, from a manufacturing perspective, there are a number of challenges that arise. Again, the first is that the plants are making a particular product for their own use. In many cases, we then have to further modify and derivatize that to get to a better medicine, and that can be very difficult, challenging, and increase the cost substantially of the product. There's a lot of risks in the supply chain and latencies, right? So it takes several years from the point of contracting with farmers to the point that you get the final product. And so if you have changes in demand with the market, or if a season of the crop fails because of climate through a particular region or pest or disease that comes through, you don't have the ability to respond to that on demand, right? And you can get then fluctuations in the amount of material that's present. If you look at it from an environmental perspective in terms of use of resources, by that I mean arable land, water, pesticides, and herbicides is actually quite costly and not a great way to be using land that could be used to make food and other products. It's inherently difficult control, right? You're growing these out in the open. And then again, access is a problem for everyone. And this feeds into the supply chains of a lot of important medicines, right? Because if you look at where plant-based medicines, what indications they're used to currently treat, it's quite diverse. They're very prevalent in the chemotherapeutic space, but also cardiovascular disease, neurological disease, infectious disease, addiction, depression, pain, cough, spasms, GI, metabolic diseases, et cetera. So the list goes on. So this affects all of those different areas, right? And so we wanted to ask, basically, many years ago as we were thinking about this, right, and thinking about what synthetic biology might enable, if we could actually use synthetic biology to shift from a supply chain that looks like this to one that basically looks like this, which is what you would see in modern pharma companies, right? And so essentially shift from growing medicines with plants to be able to brew medicines with biotechnology. And this is essentially then a high level of what the overall process looks like, right, in terms of doing this research. And it's really an intersection, right? It's at the intersection of genomics, synthetic biology, and then industrial fermentation, right? And so it starts with advances in genomics, right? And so with advances in DNA synthesis, we now have the capability to go out to the natural world and sequence many, many, many different medicinal plants. Those sequences basically hold the blueprints for how the plants are making these complex high-value molecules, right? And then we can use bioinformatic tools and computational biology to be able to search those sequences and uncover segments of DNA that we think are going to encode activities that will be useful in building out drug molecules. Now, we can take that physical information on DNA that's on our computer and then the information on DNA that's on our computer and get it synthesized into physical DNA. But this requires both advances in DNA synthesis, which we talked about as one of the core tenets of synthetic biology, but it also requires sort of the recoding strategies of synthetic biology, because if we just take that DNA and move it into a microorganism, the microorganism won't be able to read that DNA correctly. Also, if it makes the protein, oftentimes the protein isn't going to be in a functional form. So we essentially have to figure out how we recode it so that the protein can be expressed in this very different organism, and we use baker's yeast, and then we'll form a functional protein that will give us high levels of activity. And then finally, after we do that recoding and the DNA synthesis, we use tools of genome engineering to be able to take the physical DNA and very rapidly put it into our platform production strains and be able to mix different combinations of DNA, right, within a platform production strain so that we can create hundreds of thousands of different yeast strains, all with different genetic material, slightly different from one another, and then we can assay what types of chemicals they're making and at what levels very rapidly. And then finally, once we have a strain of yeast that's making the chemicals we want at the levels that we want, we have an inherently scalable platform for production. You can take those strains of yeast. They're essentially self-replicating nanofactories, right? They're going to make many copies of themselves, and you can grow them across many different volumes, whether it's the sort of sub-liter to hundreds of thousands of liters, depending on how much compound you want to produce. They'll do this over a matter of days in an enclosed facility, and they'll do this on very inexpensive media, right? And then you can recover the desired molecule from the fermentation broth. So that's what the overall process looks like. And one of the things, again, that we were interested in examining and interested in sort of the broader capacity of this type of technology was basically being able to pair the tools that I just talked about with this idea of kind of modularizing metabolism, right? And what I mean by that is that you can begin to look at metabolism and metabolic pathways in a way where you identify and define modules, right, that perform certain sets of transformations. And as you define those modules, you can begin to reuse them across different systems, right, to get to different chemistries ultimately or different final products. It has some analogies to how we think about combinatorial chemical synthesis, but now in the biochemical synthesis realm, right, and how we think about the mixing and matching of enzymes to get to new types of molecules. And so that's kind of highlighted in this slide, where each of the arrows here represents an enzyme that is performing a particular conversion. And so you can imagine that there's a set of central enzymes that allow the cell to take in sugar, convert it to an amino acid, like tyrosine, and then convert tyrosine into an isoquinoline scaffold that you see there in the middle of the cell. Through a downstream set of enzymes that you can add to the cell, it can take that isoquinoline scaffold and basically convert it to the morphin and alkaloid scaffold you see on the left. But that same core isoquinoline intermediate, just by swapping out the downstream enzyme modules, can actually be converted to a wide variety of different types of end products. And hopefully what you can appreciate in this slide is that there's a lot of diversity in the chemistry that you see up here in terms of the molecules that are being made, and a lot of bioactivity diversity, right? They exhibit a lot of different types of properties as therapeutic molecules. And so the important piece here is that as we do the R&D to basically develop out a strain of yeast that makes that isoquinoline intermediate, we can just swap in downstream modules to be able to very quickly access this diversity of chemistries. And I'll highlight that in the examples that I go over today of an example of how that's done. Okay, so now we're going to go into some examples of the work, and I'm kind of starting with the end here, or at least the end of one particular story, so that you can understand first the complexity of the system that has to be developed to be able to build out these types of natural product chemicals, right? And so in this particular example, and this particular story was published several years ago now, the idea is that we had shown that you could engineer baker's yeast to basically grow on glucose and convert it to non-natural opioids as well as nalopioids. So compounds that are not made by the poppy and that have higher value as medicinal compounds than the compounds that are directly made by the poppy. What did this take? In this particular demonstration, we had engineered the yeast with over 30 different enzymes from different plants animals, and bacteria from over 6 different organisms, right? So you see that there were sort of 4 different medicinal plants in this first example, as well as enzymes taken from rat and enzymes taken from a bacteria. And then what we're going to do is now walk through a few vignettes of how we did this work and the challenges that came up and how we had to address them, right? But this is just to sort of set the stage for, this is the level of complexity of the final system that you have to build to be able to make a chemical like this. So we're moving from recombinant DNA technology, where we were expressing one protein in E. coli, and that protein was our product, insulin, to one where we're expressing 30 plus enzymes in a yeast cell. Those enzymes are not our product, right? The product is the small molecule that's coming through the pathway that the pathway that those enzymes encode. Okay, so what are the challenges with this type of approach, right? What are the things that we had to solve? And basically, I kind of separate them into 3 different categories here, and you'll see it as we go through the talk today. But the first is that for most of the molecules we want to make, we don't know the biosynthetic pathway or the full biosynthetic pathway for how they're made in nature. So we have to go through and basically fill in those gaps, right? Do discovery of new enzymes or generate new enzyme activities that allow us to perform those reactions. The second thing is that, in particular with plant biosynthetic pathways, plants, again, have evolved to do really interesting chemistries and really, at a cell biology perspective, really interesting ways in which they regulate those processes and regulate the activity of enzymes. And so one of the things that you see very prevalently in plant cells is that they have a number of insoluble enzymes which actually have to get localized either to different compartments or within intracellular membranes for their activities to actually be present. So there's a lot of post-translational processing and modification that occurs, which is not necessarily replicated when you move that into a microbial system, and that can affect the ultimate activity of the enzyme. So you have to figure out the recoding strategy there. And again, just in general, when you have enzymes that have been evolved for activity within a particular cell type, when you move them to a different cell type, just because of differences in biochemical environment, pH, co-substrates, et cetera, that are present in that different cell type, you can have differences in activities, and you have to be able to solve for that. So a number of different challenges that go beyond the sort of, I'd say, first level of can you make the protein, right? Not sort of beyond, I would say, transcription and translation to post-translational challenges, which is sort of the next wave of challenges that synthetic biologists have to address, right, in their systems. So, again, we're going to take those as sort of basic challenges, and I'm going to just cover a few different vignettes of how they came up in this pathway. And so the first sort of piece of the story was one in which we focused first on making that isoquinoline intermediate that I showed you when we went over kind of the combinatorial biosynthesis analogy in these pathways. So this is S-reticulin, which is sort of the key intermediate to many of the isoquinoline products. And so the first thing we did was basically set up a platform yeast strain that would make S-reticulin. And again, in all my slides today, the arrows basically represent enzymes, okay, that we're putting into yeast. The colors represent kind of the source of those enzymes. So green means they came from plants, orange means they came from bacteria, and purple means they came from a mammalian system. And so right away you can see, just to reconstruct this part of the pathway, we're taking enzymes from plants, but we're also taking enzymes from bacteria and mammalian systems. Why did we do that? We did that in part because the way that plants make basically dopamine and this other intermediate for HPAA actually still has not been characterized today. So there was a gap in our understanding in this pathway. One strategy we could take was go into the plant and figure out how the plant did it. In this case, what we did was a different approach, which is to say, how do other organisms make these compounds? Because these compounds here are less specialized and are represented across different organisms, not just plants, right? Mammals make dopamine. It's a common neurotransmitter. Yeast actually make 4-HPAA. So we could look at how mammals and microorganisms were making these compounds, which had been very well characterized, take those enzymes and put them into yeast. We put all of this together, and at the end of the day, we had a yeast strain that was making fairly small quantities of reticulin, but was making quantities of reticulin. So the next thing we did was sort of traditional strategies in the field of metabolic engineering to increase production. And basically, again, each one of these is a different yeast strain that has a number of different modifications to the genetic background of the yeast strain. And what we were doing here was basically making modifications to native enzymes in the yeast pathway to increase flux to tyrosine, as well as increase flux to these building blocks. So putting feedback inhibition into some of the enzymes, as well as overexpressing some of the rate-limiting enzymes. And that gave us about a 5-fold increase in the amount of reticulin, which again is sort of standard technologies that you use in the field of strain engineering to increase production. So at that stage, we were ready to sort of look at assembling the rest of the pathway. And this is the rest of the pathway, right, as you see here. And so with a pathway this large, the strategy that we tend to take and have taken fairly successfully going forward is to separate it into modules, right? So I just talked about the first part of the module, which allows us to make S-reticulin. We split it out into 2 other modules, this middle part and then this end part. We're able to work on them simultaneously and then bring them together at the end. So I want to focus in on some of the challenges that came up with the downstream part of the pathway, which is basically decoration of the morphin and alkaloid scaffold. Okay. So this is zooming in on this part of the pathway, okay? Where the first morphin and alkaloid that's produced in opium poppy is the bane. You then have some demethylation steps as well as a reduction step that allows you to make the final end product, which accumulates in poppy, which is morphine. Now, of course, there's been a lot of interest in this pathway over the decades because of its role in analgesics, right, and pain medicines. And so there had been a lot of work to characterize the pathway and the enzymes that are responsible for this pathway in plants. So this is how we understood the pathway to occur. We took those enzymes, the 3 enzymes, the T6-ODM, the C-ODM, and the core, we put them into yeast, and then we fed the yeast the bane, and we looked at what bioconversions happened. And something very interesting, we observed something very interesting, which was we observed accumulation of morphine, but we also observed accumulation of neomorphine. And neomorphine has not been observed in poppy, right, at all. And they've done a lot of characterization on the different alkaloids that poppy makes, and this has never come up. And it was present at a very substantial level, basically a 50-50 mix between morphine and neomorphine. So it was puzzling to us because this had not been observed in poppy. Although I will say that as we've done more and more of this work, it's not unusual to see different side products and byproducts as you move enzymes from plants into yeast cells, right? And again, the reason, as we hypothesized, was that the reductase enzyme and CODM basically have activity on neopenone and neopene, so they can form this branch, right, to neomorphine. And so then the question is, okay, but why isn't this promiscuous activity observed in the plant? And we thought about this for a while, and one of the things we sort of narrowed in on and were quite interested in on was, again, the differences in how plants regulate and organize their metabolism as opposed to a single-celled microorganism, right? And what we know is that plants use spatial regulation quite a bit to kind of regulate and coordinate flux through a metabolic pathway. This can come up in many different ways, but one of the ways that this comes up is that the plant actually separates the pathway across different cell types and then shuttles intermediates across those cell types, okay? And this is known to occur in the morphin alkaloid biosynthesis pathway. It's known to occur in other secondary metabolite pathways as well with plants. And it also does this at an intracellular level across different compartments like the vacuole, et cetera. But here we were interested in thinking about how it occurs across different cell types. Now, there's different reasons that the plants might have evolved to do this. But at least a side product of this type of regulatory strategy is that you control what intermediates along a pathway an enzyme will see by separating it into different compartments, right? It's only going to see the pieces of the intermediates that are being synthesized within that particular cell type and not the ones that occurred upstream. And so what that means is that there may not be evolutionary pressure for the enzymes to evolve specificity, right? Because they're not actually seeing the full set of intermediates across the pathway. And so then when you move it into a single-celled host and have all the enzymes being expressed together, as you might imagine, they now can see everything in that pathway at once. And so if there's any sort of promiscuous activity, you're going to see that occur in terms of branching. And on one hand, depending on your objective, this could be actually a feature, right? If you want to generate diversity and look at novel products, this can actually be leveraged to do that, right? If your goal is to produce the highest amount of a final product, then it's actually a disadvantage, right? It's not something that you want. So we were thinking about this and sort of got inspired to think about how we can begin to replicate regulatory strategies based upon spatial localization of enzymes within a microorganism. And what we came to was this idea that, yeast, most microorganisms, and certainly eukaryotes like yeast, are not just a bag of molecules. There's intracellular structure in the form of different compartments, like vacuoles, mitochondria, ER, Golgi, et cetera. Peroxisomes. And so could we actually begin to leverage those intracellular compartments as a way to control the biochemical environment that an enzyme is seeing, as well as control sort of localization, right? So what we did was we created a set of tags, basically peptide sequences, that we could put onto the NRC terminus of our enzymes that would be read by yeast and allow them to route enzymes to different compartments within the cell. We validated that tag toolkit, and then we began to apply it to different problems in the pathway as we saw it come up. And so if we come back to this challenge that came up with the idea of the branching occurring, the hypothesis of why this was occurring in yeast and not plants basically came down to that spontaneous reaction, isomerization of neopenone to codenone. And the idea here was that in yeast, that spontaneous conversion was happening fairly slowly, such that core could act on neopenone and begin to shuttle it to neomorphine, right? And you could see that when both enzymes were expressed in the cytosol, we had about a 50-50 mix between the two different end products, as I had described. So what we decided to look at was what would happen if we actually physically separated the upstream enzyme from the downstream enzymes in this pathway and gave more time for the spontaneous reaction to occur as this basically product of this reaction had to diffuse to the active site of the next enzyme, right? So sort of physically separate them within the cell. Would that allow us to bias flux towards the morphine branch of the pathway? And this was sort of a different idea because there had been ideas in the field about using co-localization as a way to sort of increase flux through a pathway, so bringing enzymes closer together, either through fusions, scaffolding, as a way to, again, sort of streamline flux across different active sites. This was sort of the opposite idea of moving things apart to allow us to direct specificity towards one branch versus the other. And that's sort of what we did. So we took our tags, we basically, again, physically separated this enzyme from the downstream enzymes in the cell. And you could see that in doing so, we were able to shift specificity towards this branch of the pathway. Now, localization to different compartments gave us ultimately different ratios, right? And you can see that there were some that were approaching sort of full conversion to morphine. Certainly here, this is very close to full conversion, but we took a hit in yield. This one was because, in this case, we actually moved it into the lumen of the ER as opposed to facing outside. So we believe that there was an additional hit on yield for that metabolite to actually cross the membrane of the ER to reach the enzyme. Whereas in this case, it's sort of facing into the cytosol so that you still get you don't get a hit in yield, but still get reasonable specificity. So we ultimately wound up with going forward with this design here. But again, sort of validating the idea that thinking about the cell beyond just a bag of molecules can be really impactful and important in terms of controlling flux and specificity through a pathway, right? Both in terms of co-localization strategies, scaffolding strategies, as well as physically separating things. And so now, again, with sort of the base platform in place that allowed us to sort of shuttle flux to morphine, what we then looked at was the idea of being able to introduce enzymes that don't naturally occur in the poppy to get to non-natural opioid derivatives, right? So everything that you see here are natural opiates that are made in poppy. They're actually also made in humans naturally. And so for this, what we did is we took 2 enzymes that had been isolated from a bacterium, a Pseudomonas bacterium, that had been shown when you actually characterize these enzymes and their activity on morphine, that they had activity on the morphine scaffold. So what we did is we co-expressed these enzymes, more A and more B, in our yeast strains that were expressing these 3 enzymes with that localization strategy. And what we essentially showed is that these 2 enzymes could act on the morphine and alkaloid scaffold and produce a wide variety of different types of semi-synthetic medicinal opioids. In particular, what I'm showing you here is hydrocodone, dihydrocodeine, oxycodone, and hydromorphone, which are all approved medicinal compounds, right, used to treat different indications. And so finally what we showed here, and again, these are different strains of yeast, that we could engineer different strains of yeast that had different combinations of these enzymes, different regulatory strategies, and they would basically accumulate different profiles of alkaloids, right? And in particular, if you look at the one on the right, what we have is a strain of yeast that has been tailored to make specifically a 50-50 split of hydrocodone and oxycodone, right? And you could do further derivatizations of this to get them towards very particular final products. But this was, and why this is important, right, is that currently, if you think about the process, what happens is you extract and isolate starting material from the plant, and then you have to do several steps, in some cases many steps, of semi-synthesis to get to the final product. So, and there are certain products that are out on the market that the efficiency of those semi-synthesis steps is quite low, right? So you actually have low yield from that. So what this now allows us to do is remove the plant from the equation, right? So we don't actually have to grow anything in plants. We can make the final product in a single fermentation run. And so you both remove the upstream farming piece of it, and you remove the downstream semi-synthesis part of it, and that will substantially reduce cost, right, along the way. Okay, so we talked about the upstream part of it. We talked about the downstream part. And now we had, basically, to bring these two together, we had to focus on the middle part of the pathway. And there were two kind of remaining big challenges in terms of bringing this all together. And the first one was that there was a missing enzyme, an enzyme that had not been characterized, which basically allowed for the epimerization of S-reticulin to R-reticulin. So we had to look at that, right? And we basically got to the point where we had to kind of dig in and do discovery of this enzyme. Now, as opposed to in the early part of the pathway where I had said it's unknown how plants make dopamine, there we could take the strategy of looking at how other organisms make dopamine, right? And there had been a lot of work done on how humans and rats make dopamine, and we could leverage that to reconstruct the pathway. Here, other organisms do not make these compounds, right? So we cannot actually leverage other literature and other characterization data out there to fill in this gap. We had to go into the plant and figure out how the plant was making this compound. And this reaction of S-reticulin to R-reticulin is considered sort of the gatekeeper reaction to go to the morphin and alkaloids, right? There's sort of no other reason to make the R form unless you're going to make morphin and alkaloids. So it's really specific to plants that make morphin and alkaloids. So here we decided to take a functional genomics enzyme discovery approach. And what I mean by that is that we're not plant biologists. We don't necessarily work with the plants in our laboratories and do sort of traditional isolation. But there had been a lot of work on sequencing medicinal plants, and there was a lot of publicly available databases on genomes and transcriptomes of medicinal plants. So what we did is we took those databases and we basically began to develop kind of chemical hypotheses around what types of reactions would be responsible for converting S-reticulin to R-reticulin. So there was a little bit known in the field in terms of this intermediate dehydroreticulin had been isolated from the poppy, so it was proposed that this was a two-step reaction that went with 3-dehydroreticulin. We could look at this and say, okay, one of the steps is going to be a reduction step. Okay, so we're going to look for reductase enzymes, basically. So we could take all that genome information and basically filter out for reductase enzymes. And then what we did is we basically kind of did a negative search as well, right? So we had the transcriptome information of plants that made morphine, and then transcriptome information of plants that were related but did not make morphine. So we did like a negative search and pulled out any ones that were present in the plants that did not make morphine, looking specifically for reductase enzymes that were only unique to plants that were making morphine, basically. Now from this, there were a variety of hits that came out, okay? And basically with the tools of synthetic biology, we could actually get each one of these synthesized and just test them in yeast and see if any of them showed the activity. But when we looked at it a little bit closer, there was actually a subset, doesn't show up too well here, but there was a handful in here that stood out to us. And the reason that it stood out to us was that it was a fusion enzyme between a reductase enzyme and a cytochrome P450, okay? A plant cytochrome P450. And that stood out to us because this was the first example of a plant cytochrome P450 that was a bifunctional fusion enzyme. So it sort of was this unique occurrence of a plant cytochrome P450. And it also stood out to us because the second reaction in this pathway could theoretically be catalyzed by a cytochrome P450. So then it represented this interesting hypothesis that maybe the plants had uniquely basically evolved this fusion event to create this bifunctional enzyme, and that's what allowed them to produce the morphin and alkaloids. So we got those synthesized first, recoded for expression in yeast, put them into our yeasts that were already making S-reticulin because we had already developed that strain, and just very rapidly screened to see if any of them exhibited activity in our synthetic system. And we could see very clearly that, yes, in fact, this fusion enzyme did exhibit the activity. There's the sort of standards for S-reticulin and R-reticulin. You have an empty vector that just makes S-reticulin, and only when the fusion enzyme is present do we see conversion of S-reticulin to R-reticulin. So that was very exciting because it allowed us to finish sort of the discovery within this pathway. And for us, it was also exciting because it sort of was a nice example of the sort of shift in how enzyme discovery can be done when you leverage tools of synthetic biology and genomics, right? What I mean by that is what was interesting about this particular story is that basically within 3 months of each other, there were 3 different labs that published the discovery of this enzyme. And they each took different approaches, right? The other labs were more plant biology laboratories, and so they took more traditional approaches of creating mutants of plant lines, finding plant lines that didn't actually make the product, going in and sort of figuring out what enzymes were mutated, right, and going from that way, so really working through the plants. And this is sort of a different strategy where you get the sequence information, you start to apply chemical hypotheses to filter out, right, and create a narrower set of targets, and then you just screen them directly within your host. And you can imagine kind of systematizing this and automating this so that you can make discovery go faster, and we've certainly begun to see that in the field. Okay, so the final thing then, in terms of challenges, was that now we had systems that were making R-reticulin. The next enzyme in this pathway was this ring-closing cytochrome P450 called salsin, which also played a very important role in terms of creating the 4-ring promorphin structure. Now, it was known that this was the enzyme and this was the right enzyme, but when it was expressed in yeast, it had very, very low activity, like 1% activity. And that was really not sufficient for us to pull flux through the pathway, so we had to fix this enzyme. Now, when we characterized sort of the system on Western blots, we could see that the yeast were making the protein, and it was actually making quite a bit of it. So it wasn't a protein expression problem. But then we actually went and did a more of a careful analysis, ran like a cleaner gel, and compared it actually to the protein that was being made in a plant tobacco system, right? And what we could see is that there was a very clear difference in banding patterns, right? There's a single band that was occurring in the plant lysate, where we were seeing high activity when we fed it, the substrate. And then there was this triple banding pattern that was occurring in plants. In yeast. The triple banding pattern is indicative of glycosylation, right? And so what this indicated is that yeast were glycosylating this protein in a different way than plants. And I actually don't think that this is unusual because yeast have different glycosylation machinery, and so I think this probably occurs much more commonly than we observe, but in many cases it may not affect the activity of the protein, right? So we just don't sort of note it. In this case, it was affecting the activity of the protein. It was affecting the activity of the protein. We hypothesized, because this was a plant cytochrome P450, for its activity, it has to anchor within the membrane, right, and then be presented in the correct orientation so that it can partner with a reductase enzyme that's also in the membrane to exhibit its activity. We hypothesized that the misglycosylation was actually allowing it and resulting in it being incorrectly anchored into the membrane, so that you got very low activity. So we did a few different experiments. One is we could identify those glycosylation sites, mutate them, and show that it fixed the banding pattern on the gel, so it was definitely glycosylation. But then in order to fix the activity, what we actually took was this kind of fusion approach where we went through and basically replaced the N-terminal signaling domain of the cytochrome P450 so that we could get it correctly anchored into the membrane, keeping the catalytic domain. This created a little bit of a challenge in that we didn't know exactly where to create the fusion, right, of that chimera. We also didn't know exactly which N-terminal sequence to use, so we created a bunch of different variants where we basically went off of cytochrome P450s that were related and we knew expressed well and functioned well in yeast, took those N-terminal sequences and created different chimeras with the catalytic domain. The black is the wild type, and you could see that we were able to create a variety of different novel chimeras that exhibited increased activity. The final chimera had about a 10-fold increased activity, which was sufficient for us to bring everything together, which is what we ultimately did here. So we could bring all these different pieces together, and we're able to show, right, for the first time, a single strain of yeast that could grow on sugar and basically make two different morphin and alkaloids, the bane, which is a starting material that the plant naturally makes, and then in this case, hydrocodone, which is a medicinal opioid that the plant does not natively make, right? So we showed both of these together. Now in this first paper, the titers, concentrations that we're making were very, very low, right? It was really just proof of principle to show that this could be done, and with a pathway this complex. Since then, we have optimized the system sufficiently, right, and gotten it to titers that you would want in order to produce these things commercially. Okay, so now what I want to talk about in my remaining time is a little bit about the platform capacity and how once you do a project like this and have some of these modules set up, and also have a lot of the understandings about how you need to recode these enzymes, how you can move much more faster, right, in additional related problems. So to give you a sense, this project here, to get all of this sort of done, took about 10 years of research in my laboratory, right? And that was for a number of reasons, but you can imagine that when we started this work, the field was much less advanced than when it is now, even in terms of DNA synthesis, in terms of genome engineering, in terms of all these things. So the pace of the research, the pace of the combinations was definitely different. But another thing that really took a lot of time was really working out the challenges that I just walked you through, right? How do we recode enzymes to fix glycosylation and anchoring problems? How do we efficiently do discovery of new enzymes, right? How do we bring all these different pieces together? That was really unexplored in the field, right? And so identifying, working through this problem, being able to identify, okay, these are the problems that occur, right? These are why in certain regions proteins don't work. These are the types of enzymes that create problems. As we could begin to draw out sort of heuristic understanding about the common problems that come up and how to solve them, we could then apply that to future problems. So what I'm going to talk to you about now is how we're able to build off of this for the next project. And I talked about how S-reticulin is that isoquinoline intermediate from which a lot of different medicinal isoquinoline alkaloids are made, right? So since doing that first demonstration, we've been able to take this platform in my laboratory and show synthesis of the compounds that you see here, sort of very, very quickly with minimal effort. And I'm going to talk about one particular example, which is this example here, Noscapine, okay? And so this is the pathway for Noscapine. From S-reticulin. So everything that's highlighted in yellow is basically different than the morphin and alkaloid pathway. And you can see that it's quite long, right? There are many enzymes. So taken together, it's actually a longer pathway than that for the morphin and alkaloids. And in particular, it has many more plant cytochrome P450s, which are these ER-localized enzymes. And so traditionally in the field, it's been viewed that getting high-level expression and activity of plant cytochrome P450s is quite a challenge, right? So what you would see in the field is maybe pathways that had 1, 2, or 3 of these enzymes. In this particular example, we were able to show that we could get 7 to be functional within a single strain of yeast, right? So that represented sort of a big step forward there. Now, and all this work here was done by a single postdoc in my lab over a period of 1 year. Right, so much faster than the work that was done with opioids. So you can see again, like how getting the right tools in place, getting the right kind of framework in place allows you to then do the next project much faster. So Noscapine is interesting. I find it very interesting, first of all, from the perspective that it is a plant pathway that's encoded on a gene cluster, right? And so traditionally, it's been viewed that, microorganisms, bacteria, fungal systems, they encode pathways on these tight gene clusters, but plants have genes that are distributed across different chromosomes, and that's part of what makes discovery more difficult in plants, right? You can't just pull out one gene and then have the whole pathway there. Now, over the past decade, there have been more and more examples biosynthetic pathways in plants that are encoded on gene clusters or basically co-localized into a single region of the chromosome. Okay? And this is an example of that. It's actually one of the largest gene clusters that have been identified in plants. There's 10 genes in it. Now, of course, it's different from the perspective of that these tend to span really large genomic regions. There's very large intergenic regions, right, between different genes, and there's introns across genes, et cetera. So they don't occur in a nice tight gene cluster with a single promoter, as they might in bacteria, but they still, if you start to search across a chromosome, you can sort of pull out the different enzymes. So one of the reasons that this was interesting is that when the initial cluster was identified, that allowed us to very quickly be able to take all of those enzymes and begin to move them very quickly into yeast and begin to test their activities. And in particular, one of the things that we were able to do with the yeast synthesis platform was elucidate the final steps in the pathway, which hadn't been done before. And so really what the approach here is to leverage synthetic biology and the synthetic biology platforms to be able to advance discovery and characterization of plant pathways. And we do this via a stepwise reconstruction of the pathway. So as clusters are identified, we can basically take the plant gene cluster, recode them so that those clusters can be synthesized in yeast, so the clusters can be read and expressed in yeast, and we can basically recode them out stepwise so that we can step through what is the sequence of steps in the pathway and test that in a combinatorial fashion. Then we can just put them into our yeast strains and characterize what's being made. Again, because you can make many tens to hundreds of thousands of yeast strains, you can test all sorts of variations simultaneously and then work through what the actual steps are. Now, what was interesting about this piece of work is that at the same time, there were basically, again, 3 labs that were working on different ways of characterizing the Noscapine gene cluster. There was a lab that basically discovered the cluster in poppy and then worked on characterizing it in poppy by silencing genes and looking at what would accumulate, which is a more traditional approach. There was a lab that was taking an in vitro approach where it would express each enzyme individually in a test tube, feed it a substrate, and then characterize the product, which again is a very traditional approach. And then we were taking the approach of basically reconstructing it in yeast. So this picture, although complicated, is really meant to just highlight the results of these different strategies, with the yellow being kind of the yeast results, right? The blue being the in vitro results, and then the green being the plant results, right? One of the things that you might notice just right off the bat is that there's a lot of parallels and similarities between the yeast and the in vitro characterization, which is sort of what we would expect. They're fairly similar to each other because plants, again, because yeast don't make these compounds, you don't have a lot of enzymes that are going to be having side products on these types of scaffolds, right? So it's a fairly clean system, especially in comparison to the native plant. What you notice is that the native plant characterization is quite different. And I think the reason for that is because it's actually a fairly messy, complex system to try to do characterization in, because you can have enzymes that have overlapping activities. So when you knock one out, you might actually see then the results of a second enzyme kind of stepping in. So it doesn't give you as clean of a characterization result, right? And I think that's where these differences come in. But what we were able to show is that by and some of the importance is, I would say, especially in contrast to in vitro systems, is that by reconstructing this in yeast, we could actually get sufficient production of each of the intermediates to do NMR analysis and verification of what the structures were, which is important. And one of the challenges that comes up with an in vitro process when you're working with a pathway this complex is sourcing the substrate for the enzymes, right? Because a lot of these substrates just aren't commercially available. You can't just purchase them and make them. And so you either have to chemically synthesize them, which in some cases is very difficult. You have to try to isolate enough from another reaction. And so that can get very limiting when you're stepping through a pathway like this. With yeast, that's sort of built in as you're doing stepwise reconstruction. The yeast is making the substrate for the next step in the reaction. And so that allowed us to very quickly get towards the end steps of the pathway and be able to characterize the steps there, right, in ways that the other techniques could not. And I'll just step through what we were able to elucidate there, because it revealed some novel biochemistry that plants do. Okay, so basically, this is the end part of the Noscapine pathway, just narrowing in here, which is sort of this part, if we go from canidine to Noscapine. And so as we were reconstructing it, basically, there was still a question of how the final methyltransferase reaction in the pathway was occurring. And you can see it's the methyltransferase, the methylation of this hydroxyl group here to create noscapine. And so it still hadn't been identified which enzyme did this conclusively. Now, with the plant gene silencing study, what they proposed is, you can see there's 3 methyltransferases. There's MT1, MT2, and then actually MT3. MT1 had already been characterized to work earlier in the pathway. Okay, so it was sort of out in terms of being a suspect for this reaction. The other thing to note is that at this point is why it's believed and known that in plants, methyltransferases are homodimers, okay? So what they did was they silenced MT2. When they silenced MT2, they saw basically accumulation of narcotyline. So then they naturally sort of identified MT2 as the enzyme responsible for that methyltransferase reaction, which makes logical sense. However, when you characterize MT2 either in vitro or when we put it into yeast, we saw no activity, no conversion of narcotyline to noscapine. So then we thought, okay, well maybe it's MT3, right? So we expressed MT3, again, no conversion. So then we were scratching our heads and thinking, what's going on here? That's really the only methyltransferases that are in this cluster, so does it mean it's a methyltransferase outside of the cluster? But then we took a step back and said, okay, well maybe what explains the plant results, the plant observation, is that in this case it's not a homodimer, but it's a heterodimer between MT2 and MT3, right? And that's the active form of the enzyme. So what we did was we first expressed both MT2 and MT3 simultaneously in our yeast, making narcotyline, and we saw very clear conversion of narcotyline to noscapine, okay? And only in the case where both 2 and 3 were being expressed. We then did some in vitro experiments where we isolated the homodimers and then isolated the heterodimer, and again showed very conclusively that it's the heterodimer that has this activity and not the homodimers, right? So this was interesting for 2 reasons, right? First, it allowed us to complete characterization of the pathway, but also I think it was the first example of a heterodimer methyltransferase catalyzing a reaction in a native plant biosynthetic pathway. So it really then highlighted that heterodimers are probably occurring more frequently than we expect and are responsible for different methylation chemistries within the plant that we haven't yet identified. And so as we do characterization of enzymes and characterization of these pathways, we should also be looking at heterodimers and not just homodimers of these genes. And so I think that's an important takeaway there. So then ultimately, with this platform, as I said, we were able to bring all these enzymes together in a very short period of time and basically show production of noscapine from a single yeast strain grown on sugar. There were a number of other sort of optimization strategies that applied, and that allowed us to boost titers up further from what we had seen from the morphin and alkaloid pathway. And so I'm going to start to wind here. But I just want to make a few different comments, right, about the technology and how we might think about it. So one of the, I think, good things that comes about from doing work like this is that there's a lot of discussion about the implications, right, of technologies that are basically making controlled substances, which is what the medicinal opioids are, right? And whether that's a good thing or a bad thing and how that can be used, right, in different contexts. And so one of the things I would like to highlight here in terms of how we think about it is that we think about this technology really as a way to offer more control into the supply chain, right? So it's not really about having a technology that allows us to make more of these compounds, right? It's a technology that allows us to make these compounds in a more controlled way, in a more cost-affordable way, so that there can be better access generally and not isolated to a couple countries, right? And also a technology that allows us to more efficiently make higher value, better medicines, right? So again, what are the differences if we think about what's currently the method and what this new way would allow? The first is that this method of reconstructing pathways in yeast allows us to precisely fine-tune the molecules that we make, right? So we're not just inheriting what nature has given us in terms of a morphine product, right? But we can decorate and derivatize that scaffold using enzymes very efficiently to be able to make higher value compounds that have less addictive properties, right? And that can be used to treat substance abuse, which I think is really an important application of the technology. The second thing is that, again, from a control perspective, it allows us to remove the risk and latency in the supply chain, right? And why is that important? I mean, it's important for a number of reasons, certainly from a manufacturing perspective, but it's also important because it moves from sort of a system that has a lot of latency, where you might be pushing product into the market, right, because you cannot respond very quickly to changes in market demand, and what you've sort of either scheduled or contracted to produce 3 or 4 years ago is sort of what's coming out, right, at that time, to one where you can fire up fermenters on demand and be able to respond more quickly to changes in demand that occur within the market, so that you're pulling product in based upon demand as opposed to pushing based upon what you've sourced. And the third thing, again, comes down to what synthetic biology can allow, which is, again, here you can imagine, and I think appreciate, that it's an inherently uncontrolled supply chain, right? You're growing these out in the field. Farmers have to deal with this. There's a lot of challenges, right, in terms of control, diversion, et cetera. The synthetic biology approach has a lot greater control built into it. First of all, you moved everything into an enclosed, contained facility where you can track what's being made and where it's going. And in addition to sort of the physical control barriers that are there, you also have genetic control barriers, right? Using tools of synthetic biology, you can actually engineer control modules into your yeast so that somebody cannot just take the yeast strain and grow it up in their own system, that there can be proprietary chemicals and inputs that have to be fed in order to get the strain to produce, right? And different mechanisms like that, again, which actually build in greater control against possible misuse issues. So I think all of these are sort of how we think about the value that this can add, right, to existing supply chains. And then, again, if we just take a step back and think about this more broadly, because I showed you in this example, starting with what I would call sort of a very substantial challenge in the field, knocking that off in terms of being able to make the morphin and alkaloid scaffold, and how doing that really positions us to be able to work more quickly on the next project and then the next project. So you can imagine this becomes a platform for really being able to produce many diverse plant alkaloid medicines very efficiently, and then the impact that this has over time. Because again, the central role that small molecule therapeutics and plant natural products play in medicine and healthcare. And I'll just leave you here, again, with just some things that we might want to think about as we think about applications of this technology and how we commercialize it and how we make it available going forward, right? And so things that we think about really, right, in terms of the value that we're creating is how do we use these new technologies, what synthetic biology really opens up and enables in terms of being able to move from systems where we're engineering a single gene into a recombinant organism to engineering 30 to 100 genes into a recombinant organism. What does that give us in terms of new power, new applications, and how we can use this to sort of better the human experience, right? And can we develop partnerships, right? And I think this is important too, right? Is to think really about how this type of deep technology partners with our natural world, right? And actually creates both a healthier people and a healthier planet. You can think again about the environmental consequences, right? Is it really a good idea to be using arable land and water and all these pesticides and herbicides to be trying to grow medicinal plants where we're throwing away a lot of the biomass, right? Or we're over-harvesting and losing biodiversity. If we can shift to this type of production platform and really use our arable land for growing food, right, that can be sort of a big win there, and keeping diversity within the planet. And then finally, as we think about bringing this system into commercialization, right, what are the new systems and structures and partnerships that really need to be developed so that we can really address some of the access issues that are prevalent sort of globally, right? And it's not just available to a small fraction of the population. And that's the end of this talk, so thank you all. Happy to take questions, and just thanking the people that have been responsible for the work. As I said, the work that I shared today was done over many, many years, and so there were many graduate students and postdocs who played central roles in driving this technology, and the work and the successes are really all of theirs. So thank you.
It's a very impressive talk. It's amazing that you can make scientific discoveries as well as engineer yeast that can basically make drugs. I'm just curious, although it's very amazing that you can physically scramble 30 genes in one yeast strain and to achieve from very beginning to the end of this entire synthetic pathways, But alternatively, whether it's possible to basically make different modules in different yeast strains and optimize it for each of the part, and also some of the part can be combined with chemical synthesis.
Yes.
Because that might be more efficient in some intermediate, and then basically make some kind of decision labor.
Yeah.
Making it easy to scale up.
That's a very good question. And so the question is around, how do we think about the trade-offs between trying to do this all within a single yeast strain versus, one hand, separating the pathway across multiple yeast strains or a yeast and bacterial strain, et cetera. So let me address that part first, and then we talk about the kind of semi-synthesis routes. So I think the idea of splitting across different strains is an interesting one, but then you have to think about practically how that scales, right? So one, because you're talking about going from something that's maybe sub 1 liter, a shake flask or whatever. Ultimately, this is going to be, if you're going to commercialize it, you're going to be doing this in a huge reactor of 100,000 liters, right? And so there's a few different ways that people envision that happening. One way would be a co-culture, where you're still doing one fermentation, but you are growing multiple strains within a single fermentation, right? I think that, number one, is very challenging to control at an industrial level because it's very difficult to control the relative growth of the strains, right? You're not going to use antibiotics or oxytrophies or these other things to try to control growth rates as you scale commercially. That's going to be too expensive. So you're really just going to seed the thing and, kind of see what happens. But it's going to be very challenging to get the growth rate balanced correctly and get that to be reproducible, right, as you go to large industrial scale. So I think that's the first challenge there, thinking about co-cultures at industrial scale and how you control that. And then the second challenge that I see for these products in particular is that the transport of the molecules into the cell is very inefficient. The yeast are very good at pumping them out. Okay, that's what they want to do. They have multi-drug resistant pumps that are just getting these things out of the cell as quickly as possible. That's why also it doesn't really affect the growth of the yeast because there's not very high concentrations intracellularly, right? But it's very difficult to get that alkaloid scaffold back into the cell. So if you try to do something which depends on transport across multiple cells, you actually lose a lot of flux, right? Just because things aren't moving into the cell. So that's the other challenge that you have to deal with. The other option might be do multiple fermentations, right? So do one fermentation with the first part of the pathway, use that substrate to feed into a second fermentation. And again, there I think you're going to have a cost issue, right? Because every fermentation you run is basically a process cost, right? And that is just going to add substantial cost to these overall processes, and I think is going to be difficult to work commercially. You really want to minimize the number of fermentations you have to run if you're going to get this to work at a commercial scale, right? You'd also have to purify the product from the first one, and that's also going to cost. So I just think that becomes problematic for those reasons. There are some chemicals that aren't of these types of drug structures, which actually move into cells better. And that there's sort of the examples that we've seen in the academic literature of people showing these co-culture systems, right? Those, then you don't have that barrier. So I think it also really depends on the structure of the molecule and how well they get across cells or imported into cells for that piece to work, right? But for these types of drug molecules, it's very challenging. Now, your question about medicinal synthesis or chemical synthesis is a very good one, and I agree with you. And I guess what I would say again is this. There are some reactions that biology is not good at, but chemistry is very good at, right? And then there's no reason to basically try to spend a lot of money, a lot of dollars to evolve an enzyme, that's going to try to do that reaction. It just doesn't make a good kind of cost benefit analysis at the end, right? And there you can absolutely do processes where you make, a starting material, you do all the difficult kind of chemistry in the cell, get to a product that's maybe 1 or 2 steps away from the final product and do semi-synthesis at the end. There are some reactions that biology is going to be very good at that chemistry is not. Those are definitely the ones you want to do in the cell. There are some that both can do. And again, I would just say if both can do them well and you can move it into a single process, you'll get a cost advantage. So you just want to weigh all those different things in terms of how it ultimately feeds into the cost of the product. But I do agree with you that there's really no reason not to use chemistry on the back end if it makes sense from a cost perspective. Yeah.
Talk about this 10-year project. When you came up with this idea in the first place, how many years have you prepared to or planned to?
How many years? Yeah. So that's a good question. I mean, so basically when I was starting my lab at Caltech, I was doing what a lot of assistant professors do, which is thinking about what I wanted to do for my research program. And, I was thinking about sort of big challenges, right? And so for me, there were a couple things, right? I wanted to, typically you're thinking about 10-year challenges, right? What am I going to do over the next 10 years? And what are kind of the big challenges? And so, again, for me, what I was thinking about when I was defining this was, I had done my training in metabolic engineering at the time, which is, several decades ago. And so then the sort of state of the art was working with maybe 3 or 4 enzymes, right? And that would be kind of really impressive, right, in the field. And that got you to, a very small subset of molecules, right? And so, again, from the background in chemical engineering and thinking about synthetic biology and everything else, so to me, when I set up my lab, it was, what are the things that are currently now impossible, right? And this was one of those projects, the idea of engineering yeast with 30 plus enzymes. And I can tell you, when I submitted grants, that's what I would get. It's like, this is impossible. Yeah, don't waste your time. It was actually very difficult to get funded in the beginning, right? Because it was just so far out there, that people would just say, this is a waste of time. Like, you can't do this, right? And for me, it was like, that's exactly why we should be doing this, right? Because what I want to do in my research is I want to be working on problems that are currently impossible. That's what we should be doing in academia. And that also fuels, from a synthetic biology perspective, what tools we need to be developing, right? So that was the other thing, is that I had a very strong interest in tool development, which sort of comes through some of the talk, right? But I felt like it was very important to do tool development in the context of application, and not just tool development in the context of, look at this, right? Because I wanted to understand, okay, if you're trying to work on these problems, where are really the bottlenecks, and what things don't matter and actually what things do, and where should we be spending our time on the tools, right? So that was sort of all what came about in terms of defining the problem. And then, the real idea here was, let's start with the thing that's the most challenging. And if we get that to work, then it's going to be this thing where we've worked out all the problems and we think about it very generally in terms of defining general problems. We can apply that to the next one and the next one, right? And then it's just going to be this sort of cycle, which, and what I see is sort of what's happening, right? So that was how we thought about it then.
Have you ever worried, whether you can really accomplish or complete the entire?
Yeah. Have I ever worried? Yeah, of course.
I think that a decade ago, sounds like a mission impossible.
Oh yeah, even 5 years ago, people were saying it was going to be impossible, right? And it's funny because even after the paper was published, then people said, oh, you'll never get, this is an interesting proof of concept, but you'll never get it to be commercially relevant. And at that time, I was like, no, it's done, right? It's gonna be that in like 2 years, and it was, right? Because they just didn't understand where the technology was, right? And that once you get that pathway and that proof of concept, it's very now routine to just sort of push on the titers, right, and the yield and everything else. There's a set of tools that allow you to brute force that. But yeah, so I think it's something, what I find, right, and I think in academia, we should be working on the hard problems. We should be working on the things that sound crazy and like people are just like, and basically are not going to be done in industry, right? Because they're too far out there, and we should be pushing on it. And it's, and again, I think from my perspective, when I thought about it, I think, I always believed that we could get there, right? If plants can do it, we should be able to reconstruct it in a microbe, right? Like, we just have to figure out what the problems were. But in order to justify, I guess what I would say again from maybe like thinking about how we organize our labs, what I had to justify for myself and also for the students working on this was that we didn't have to do everything to declare success, that we could break it up into projects that graduate students could work on along the way, that could be published, that would be significant contributions, and that it wasn't just like, we have to get the full system in place to do anything, right? Because that would have been very difficult to do in an academic lab. So I think that's the other thing, is like when we define these really large problems in academia, we then have to really think about how we break it into chunks and how we can declare success along the way and build upon it. Yeah.
Okay,
So I think it's a very nice concluding remark to many of our PhD students who are working on a project. Try to find, don't be too discouraged when you have hard problems.
Oh, yeah.
So I think we would like to conclude this presentation with a high note. Please join me to thank Christina again.